Hey everybody, welcome to chapter 17 of Human Anatomy and Physiology, the Marieb and Hone textbook. This chapter is on blood physiology, and I have this profound quote here from your book, "The river of life." A little dramatic, sorry, but pretty accurate, I suppose. So we're going to talk all about blood chemistry and the composition of whole blood, and we'll talk a bit at the end, quite a bit at the end actually, about transfusion reactions. I have a tendency on my tests to do a lot of transfusion reaction questions. If you give type AB blood to someone with type O blood, what happens? Are you in trouble? Did you save the day? That kind of thing. So um, I'm anticipating this being fairly short, about an hour. So let's get to the chase. All right, so blood is unique because it's the body's only fluid tissue. There are other fluids in your body, interstitial fluid, which is the extracellular fluid around cells, intracellular fluid, but none of those fluid compartments are a tissue in its own right. Blood is the only one. And blood is actually a connective tissue. It has both cellular and liquid components, just like connective tissue has cellular and extracellular matrix, which is more viscous than blood, but nevertheless pretty similar. Um, in the case of blood, we have cells, cell fragments, and cell-like structures, like red blood cells, suspended in a fluid called plasma. And that plasma is kind of the equivalent to an extracellular matrix, which if you weren't here last quarter, that's just something unique to connective tissue, is that there's this large space, a large percentage of the tissue isn't cellular. It's something else. It's a matrix. And in the case of, areolar or connective tissue, it's a viscous water-based matrix with different types of proteins that make it viscous. And in the case of blood, it's plasma. In the case of bone, it's a calcified collagen matrix, collagen and protein matrix. So every connective tissue is different, but the fact that there is an extracellular matrix is something that is unique to connective tissue. Blood also has a mesenchymal origin, which is something also unique to connective tissues. The cells within connective tissues have a mesenchymal origin. So what is blood made up of? So 45% of it is erythrocytes. This is the most common formed element found in blood. So you can see there's two parts to blood: formed elements and then plasma, which is what the formed elements are floating around in. So erythrocytes is most of that, about 45% in a healthy adult. So not anemic, not polycythemic. That's kind of the standard normal value would be 45%. And there's a range that's considered healthy. This is kind of the average. This represents the oxygen carrying component of your blood. Some oxygen can be dissolved in plasma, more carbon dioxide, but some oxygen can. But for the most part, this represents the oxygen carrying capacity of your blood. So if this is really low, your ability to carry and transport oxygen to your tissues is going to be diminished. If it's really high, you're either in really great shape or you're taking some bad drugs or you live at a really high elevation, who knows. But it can be normal to be elevated, but it can be abnormal as well. Just like it can be abnormal to be low, which would be called anemia. If this value is pretty high, that's called polycythemia. And we'll talk about what might bring about those conditions a bit later. So anyway, those are the red blood cells, the erythrocytes. That measure the percentage of your blood that is made up of erythrocytes is called the hematocrit. So a common thing you might read in a hospital chart is H&H, hematocrit and hemoglobin. We'll talk about hemoglobin a little bit later. The other formed elements are leukocytes and platelets. So these are white blood cells and platelets. These make up less than 1% of whole blood. And there's this little thing here called the Buffy coat. It's this white line that divides the plasma from the hematocrit. Sorry, very small amount as a percentage. And then the remaining 55-ish% is plasma. and then together that's whole blood. So you can cause blood to separate like this if you put it in a centrifuge, spin it around, and the different components separate by density, and you're left with this vial that you can kind of look and see if the percentages make sense. Okay, so what does blood do? I think most of us know it's a distribution mechanism. It carries oxygen to your body's tissues. It carries CO2 from your body's tissues back to your lungs, delivers nutrients. It carries waste to the liver to be metabolized, to the kidneys, to the lungs, and then it's a hormone transport mechanism. So distribution is kind of the main thing that we sort of all know intuitively to be true. It also regulates temperature. Basically, if it's really hot out, all of your precapillary sphincters, so you have these little smooth muscles that control how much blood is going to various capillary beds in your body. And your skin has a lot of capillary beds in it, your dermis. So if it's really hot, your body can dilate those precapillary sphincters and bring a lot of blood to your skin, to your dermis. And that causes heat to radiate from your body. And, assuming it's less than 98.6 degrees, it works really well. Still works, in the hundreds, but it's just not quite as efficient. Sweat plays a big role, so you sweat on your skin, and the heat radiates and heats up that water, and the water evaporates and takes the heat with it. And so that's a way that you can regulate heat. If you're really cold, your body will stop the blood flow to the skin. It will actually allow your skin to become the same temperature as your surroundings, which is why if you're in a situation where it's below freezing, your body will actually allow your skin and your distal extremities, your fingertips and your toes to freeze, literally, not just like, oh, I'm freezing, but actually freeze. And so all the fluid within your cells freezes, and you guys know water expands when it freezes, and it causes the cells to lyse and break, and that's frostbite, or I guess frostbite. I was looking for a better term for that, but that's pretty accurate, I guess. So so it also regulates pH, so what the acidity of your blood is. That's done in a few different ways. There are some buffers in your blood that regulate pH. Also, your lungs, your breathing rate influences pH. You can breathe faster, which will bring more oxygen in and remove carbon dioxide. That will make your blood more alkaline. And if your breathing rate slows, that makes it more acidic. So your blood regulates temperature, pH, and then fluid volume. Your fluid volume is regulated in many different ways. So renin and aldosterone mechanism in your kidneys can cause you to retain water and maintain fluid volume and therefore blood pressure. Antidiuretic hormone, vasoconstriction of your arteries influences pressure, not so much volume, but pressure. So that's critical. Your blood pressure needs to be within a certain range in order for blood to be delivered adequately and appropriately to all of the different tissues in your body. So temperature, pH, and fluid volume are all critical. And then the blood also has a mechanism for protection, really protection from its own loss in the form of clot formation. So if you cut yourself, there's a process that we will look at called coagulation that causes that blood loss to correct, depending on how severe the wound is. But in most cases, small scrapes and cuts and abrasions and things like that can easily resolve themselves through coagulation. Then you also have antibodies in your blood. So antibodies are a part of your adaptive immune system. And we're not going to get to the immune system in this quarter, but antibodies are a big part of that. Then we have various other white blood cells, which are also a part of your immune system. Okay, so let's go look at plasma. So plasma, remember, it's 55% of your blood. and that 55% of your blood is 90% water. And within that water, we have about 100, actually more than 100 different solutes, which are nutrients, gases, hormones, waste, proteins, ions. And then of those proteins, about 60% of the plasma proteins are albumin. Albumin and these other plasma proteins are not fuel. Well, some proteins that are delivered to your tissues are, but albumin specifically, this is not a fuel meant for consumption by your body's tissues. Instead, albumin maintains plasma osmotic pressure. Now, osmotic pressure is can be a little confusing. Why can't I draw? There we go. Let's say this is a capillary. Whoa, ignore that. That's crazy. Let's say we have a capillary here, okay? A skinny little capillary filled with blood. And it's going, let's just keep drawing it like this. Well, capillaries, most capillaries, except for the ones in your brain, are very leaky. So there are cracks that fluid can just kind of slip out of. And honestly, if I wanted to get super, super technical, you could almost draw capillaries like this. So where the epithelial cells overlap, there are little gaps. This almost looks more like a vein with one-way valves. That's not my intention. So these are little endothelial cells, and there are little gaps between them. And there's gaps here, but then there's also gaps, in between the adjacent walls of the cells, and so they're leaky. Fluid can leak out. So say the pressure here is, depends where we are in the body. Let's say it's 80 millimeters mercury. Okay. And then let's say right here it's 75. So fluid is going to leak from your circulatory system into your tissues. Okay, so this is into your tissue fluid, your interstitial fluid in between your cells. Now, let's say, let's say we're not really in a position to lose 5 millimeters mercury, because given enough time, if this is 80 and this is 75, with time we're going to reach equilibrium. So this might become 76 and this might become 76, depending on all the different volumes, and there's all different types of variables and factors at play. But eventually the pressure would decrease here. But maybe we don't want that. Maybe we want to maintain this 80 millimeters of mercury. Well, we need some kind of force pulling fluid back in to maintain this equilibrium. And that is osmotic pressure. So hydrostatic pressure is the pressure forcing the fluid out. Osmotic pressure is the pressure pulling it back in, and it acts like a sponge. So if we have a bunch of albumin inside our circulatory system, inside this capillary, albumin is too big to leak out of these gaps between cells. So it acts like a sponge and pulls the water back in. Sodium could do the same thing, but the difference is sodium could easily leak out. It's a tiny little ion. Albumin is much bigger. So all solutes would create osmotic pressure potentially, but the difference is albumin is large and it can't leak out into the tissue. So it stays in the blood, meaning it pulls that back in. So you have this tug of war between hydrostatic pressure and osmotic pressure. So anyway, I don't ask a ton of questions about that typically, but if I do, it's usually along the lines of, plasma proteins like albumin, do they serve as a fuel for cellular metabolism or maintaining osmotic pressure? And so they maintain osmotic pressure. There we go, boom, there's the question. I'm so transparent. Oops, sorry. Okay, so let's talk a bit about formed elements now. Ooh, boy, we got a few more slides than I expected. So formed elements are things floating around in blood, and not something dissolved like a solute, but something with formed element. So there are three types of formed elements: red blood cells or erythrocytes, white blood cells or leukocytes, and then platelets. Two of those are actually not even cells. Red blood cells are not cells, which is a bit confusing because they're called red blood cells. And then platelets are fragments of these really large cells called megakaryocytes that are within your red marrow, within your bones. So the only one of these three formed elements that are actually cells are white blood cells. Most of these don't last long in the blood. A few days to a bit longer. There's just a range. And most of these do not divide. So let's talk about erythrocytes first. Um, I don't know if any of you guys are from the Midwest. I grew up like, I guess 10 years, like 4 years old to, or like 3 years old to 11 years old, something like that, um, in Michigan. And so a big season was fall because it meant like winter was coming, and winters in Michigan are different than winters here. Winters here are like, oh no, it's going to be 50 degrees, but in Michigan it means every morning on the way to work you have to scrape the ice off your windshield, and you have to warm your car up for an hour before it will move out of its frozen pile of snow. It's different. They're harder than here. Anyway, so fall was kind of like your last chance to go outside without freezing to death. So they had like not pumpkin patches. We have pumpkin patches here, but they had like these. But you could get pumpkins there. So yes, they were pumpkin patches. But they had like it's kind of like Oak Glen. If any of you guys have been to Oak Glen, it's like Oak Glen everywhere. They're all over the place. You can get apple cider and pumpkins and all different kinds of stuff. And one of the things they had were these little donuts that you dipped in the cider, and they were not very good, but dipped in cider, they were really good. I can't believe I'm talking for 5 minutes about this. I'm sorry. I apologize. But I just always think about it when I see this slide because they look just like red blood cells, which brings me, I always tie it back to the shape of the red blood cell because they are a biconcave disc. And that's not by accident. So basically, if red blood cells were a sphere, I know this is a circle. Well, that's not even a circle. I don't know what that is. But okay, anyway, say they were a circle. It's a little bit better. Their job is gas transport. Red blood cells live to move oxygen around and carbon dioxide to a lesser extent, oxygen in particular. So they're filled with this chemical called hemoglobin, which binds to oxygen in a manner that makes it ideal for oxygen transport because it picks oxygen up in an oxygen-rich, cool, uh, high pH environment like your lungs, and it delivers it to low pH, hot, high carbon dioxide environments like tissues that need oxygen. So it's perfect. It's a great way to shuttle oxygen chemically. So anyway, say this was a real red blood cell and you have hemoglobins all over the place, right? Hemoglobin, 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 hemoglobin. It'd be much easier for this hemoglobin to drop off oxygen than this hemoglobin, right? Because this hemoglobin is much closer to the cell membrane. So you don't want that in a red blood cell. You want no hemoglobin molecule too far from the cell membrane because you want readily easy gas transport. So the way to do that is to make the red blood cell a biconcave disc because when it's shaped like that, there's really, and that's a bad drawing, there's no part that's too far away, okay, from the surface. So it's easy for hemoglobin to pick up oxygen and deliver it. Anyway, red blood cells are anucleate and they have no organelles. So they're literally a bag of hemoglobin. They're 97% hemoglobin. I said Hg, I guess I should have said Hb, sorry. They're highly deformable, so they can move through very small capillaries. And they generate ATP anaerobically because number one, there's no mitochondria, but number two, you don't want to consume the oxygen that you're trying to transport. It'd be like, those big trucks that haul gas around the country. It'd be like if they used the gas out of their trailer. By the time they got to their destination, it'd be like half full, right? So you wouldn't want that. So they generate ATP anaerobically, and they're basically just hemoglobin, cell membrane filled with hemoglobin. So what do? Well, we've kind of already alluded to that. Gas transport using a molecule called hemoglobin. So hemoglobin is made of these 4 chains, 2 alpha chains, these are protein chains, and 2 beta chains. That's not particularly important. What's important is the heme group. So at the center of each of these chains is one heme group. So there's 4 total. You can't really see the other 2 in this photo, but there's 4 of these. And at the center of each heme group is an iron atom. Okay, and that iron atom is capable of oxidizing. Okay, so what do you think I'm getting at here? So each hemoglobin molecule is capable of picking up 4 oxygen molecules, 4 O2s. So the oxygen, the O2, binds to the heme group, specifically this iron atom, and then it becomes oxidized. And when iron oxidizes, it turns red, which is, if you've ever seen rust, that's iron oxidizing. So the heme groups quite, literally rust within each of your hemoglobin molecules, meaning they turn red. That's why your blood is red when it's oxygenated, or it's a big part of why it's red when it's oxygenated. Um, and so this hemoglobin molecule then can carry oxygen. And when it's oxygenated like that, it's called oxyhemoglobin. When it's not oxygenated at all or partially oxygenated, it's deoxyhemoglobin. And then when it actually has carbon dioxide bound to the chains, because carbon dioxide is capable of binding to these alpha and beta chains, it's called carbaminohemoglobin. That said, most hemoglobin is actually most carbon dioxide is just dissolved in your plasma, or in the cytosol of the red blood cell. But the globin chains are able to pick up some carbon dioxide. Anyway, so the point of this is that hemoglobin is responsible for picking up the oxygen molecule, specifically the iron atom in the heme group. Now there are 4, so that means that one hemoglobin molecule can hold 4 oxygen molecules, and there's about 250 million hemoglobin molecules in each red blood cell. So some quick math there. That means there's about 1 billion oxygen molecules per red blood cell. I usually ask a question like that, so just a heads up. Production of red blood cells, we don't go into huge detail here, but basically this process is called hematopoiesis. It's the process for making all red blood cells. This process occurs in red marrow. Man, look at that. 100 billion cells a day are made. So if you're ever feeling lazy, just remember that day you made 100 billion cells. That's pretty cool. You're pretty impressive. All blood cells arise from a hematopoietic stem cell. So you'll see if you continue your studies of physiology, stem cells are very interesting. You have what's called a multipotent stem cell or an omnipotent stem cell. That stem cell can become just about anything. And what you'll see in any tissue that's responsible for producing new cells is that there's some further differentiated stem cell. So in this case, it's a hematopoietic stem cell. So this stem cell can give rise to all the other blood cells. In bone, you have the osteoprogenitor cell, which is a stem cell that's just a little bit more differentiated than a multipotent stem cell. But anyway, it's all you need to know is that red blood cells come from hematopoietic stem cells. I'm sorry, not just red blood cells in general. Come from hematopoietic stem cells. Erythrocytes in particular come from hemocytoblast, which is an even further differentiated stem cell, which turns into a proerythroblast, an early erythroblast, a late erythroblast, a normoblast. And then once the nucleus is ejected, they become a reticulocyte. And reticulocytes are present in your blood, not just in the red marrow. So one way you can tell if someone is, let me phrase it differently. So let's say you're worried someone might be bleeding internally. I'm sure there's much better ways to figure this out, but let's just say you suspect that and you want to figure it out from a blood test. Well, if someone is bleeding internally, they're probably anemic because they're losing blood, right? So if someone's anemic, meaning if they're low on hematocrit, low on red blood cells, their body's probably trying to account for that, to accommodate that, counteract that by producing a lot of red blood cells so you can carry more oxygen. So what that means is that there's going to be more reticulocytes than normal in your blood because this process is happening at a greater scale. So there's going to be more reticulocytes. So if the normal reticulocyte number is 1 to 2% of erythrocytes in someone who's healthy, and you do a test and see that 15%, I'm sure that's probably too dramatic, but 15% of their red blood cells are actually reticulocytes, you're going to suspect some kind of issue, some kind of anemia. bleeding internally, sickle cell, thalassemia? No, I'm sorry, that's not. No, thalassemia. Yeah, sorry. That's a different type of anemia. So it's just kind of an interesting little nugget, I guess. But anyway, this is just the basic steps of the process of producing erythrocytes. And that's regulated hormonally. So you produce about 2 million of these guys every second. And basically, your kidneys are capable of detecting the oxygen carrying capacity of your blood. So if they detect a decrease in oxygen carrying capacity, your kidneys release something called erythropoietin, or EPO, into the blood. And EPO stimulates what we just saw on this last slide, erythropoiesis. And it has a negative control loop because as soon as you stimulate erythropoiesis, erythrocytes are going to be released into the blood, thus increasing the oxygen carrying capacity, meaning that EPO will stop being released. Because remember, EPO is only released when a decreased oxygen carrying capacity is detected. So it's this little negative feedback loop. So this can be abused. This process can be abused, and I think it's coming up on the next slide. But there is something called blood doping, and you can do that using synthetic EPO. We'll talk about that in a second. Okay, so now we're going to talk a bit about erythrocyte disorders. And erythrocyte disorders basically take one of two forms: too many or not enough. Anemia refers to not enough. So either there's not enough red blood cells, I apologize. It is due to there not being enough red blood cells, but that's either through blood loss or some type of problem with blood cell production or some type of issue with destruction of red blood cells. Blood loss is pretty straightforward. So you have a wound or an injury that's causing you to bleed internally, and your blood volume is going down, and therefore you have less circulating red blood cells, or it could be due to not enough cells being produced for a variety of reasons, iron deficiencies, some other type of deficiencies, other disorders, or too many cells destroyed. Two examples of that are thalassemias and sickle cell anemia. These can happen in any race, any ethnicity. However, thalassemias are much more common in people of Greek ancestry, and sickle cell anemias are much more common in people of African ancestry. In either case, there's some type of issue with a amino acid sequence due to a particular gene coding for those codons that make up that peptide incorrectly. So one particular codon, and I apologize that I don't know the specifics, but one particular codon is codes for slightly differently, and that causes the cells to be shaped abnormally. And because of that, I'm so sorry. And because of that, your body destroys those red blood cells because there's something wrong with them. And that leads to anemia. So any of these things can cause anemia: blood loss, not enough cells produced, or too many cells destroyed. The opposite of anemia is polycythemia, so too many cells. This can happen for two reasons, or there may be more, but the two most common reasons is polycythemia vera, which is a type of cancer of the red marrow, which causes way too many red blood cells to be produced. And then the other is blood doping. Blood doping is something that that you do to yourself. So it's unnatural in that sense. So this is common in sports that require tremendous aerobic capacities, like triathletes, marathoners, cyclists. I think Lance Armstrong, this is ultimately what he got caught for. I could be wrong. It might be something else, but there's all different types of performance enhancing drugs and things. Blood doping is a very specific method of increasing your hematocrit. And the easiest way to do that would be to take EPO, synthetic EPO, erythropoietin, because that stimulates red blood cell production. And then you have more red blood cells, which means you can carry more oxygen, which means you can ride your bike farther. Oh my goodness, I'm yawning like crazy. I need some oxygen in my blood. Anyway, the issue there, and in addition to it just being potentially dangerous and unhealthy for you, the issue there is that that's detectable. So someone could simply measure your EPO levels before the Tour de France, and they would detect that your EPO is much too high for someone in good shape, and therefore you're busted. So there's other types of blood doping that are very dangerous but undetectable because there's no actual drug that you take. Instead, what you do is you give blood, you take blood, so you have some trusted individual draw blood from you, and then you go train and you go stimulate your body to compensate and replenish those red blood cells that you gave. And then you take some of the plasma out of those red blood cells and then you put them back into you. So you basically just do blood transfusions on yourself. That could be someone from someone else with the same blood type as you or your own blood. Either way, it's potentially dangerous. You can think about, there's a certain point where it's unhealthy for your hematocrit to increase any further. You can think about how the viscosity of your blood might be affected and what kind of toll that could take on your kidneys. And anyway, but the point is that would be undetectable because all you've done is inject yourself with your own blood, and there's no way to detect if you have your own blood in you, because of course you do. This is really just a simpler way and a quicker and more dramatic way of doing high altitude training. For example, you go up high into the mountains, and then all of a sudden your body is oxygen poor, so your body releases EPO, and your red blood cells, your hematocrit increases, and then you can come back down to elevation, to normal elevation for a race, and you have an artificially high hematocrit, and your VO2 max is increased, your aerobic capacity is increased, and you could perform better. So anyway, so just two different types of erythrocyte disorders, two different categories. You guys know this. 4 molecules of oxygen per hemoglobin molecule, and it binds to the iron atom within the heme group. Okay, let's look at white blood cells now. So white blood cells are true cells. They have a nucleus. They carry out cellular activities, and they have organelles. For the most part, they are produced in the red marrow, so they aren't necessarily undergoing a lot of mitosis like other cells, but they are true cells. They carry their own genetic material, so they're different from red blood cells and platelets in that way. A normal amount is about 4,000 to 11,000 per cubic millimeter. So more than that is leukocytosis, and that leukocytosis can be specific to a certain cell type. For example, if in particular it's neutrophils accounting for the leukocytosis, in particular that might be called neutrophilia. So there are different types of leukocytosis, but more than 11,000 per cubic millimeter. That might happen if you have a severe infection. I mean, you might have 100,000 per cubic millimeter if you have a severe infection, or cancer is common. For example, if any of you have a family member or relative that has survived cancer and is in remission, they most likely go back to their doctor, every 6 months or so, and one of the tests they will do is a white blood cell test, because something that is associated with cancer is elevated white blood cell counts. Anyway, so there are two major categories, granulocytes and agranulocytes. And this just refers, you can look at this picture here and probably guess which of these are granulocytes and which are agranulocytes. Granulocytes have visible stained granules under a microscope when the slide is stained. And that's due to the fact that there are all of these little vesicles inside these cells. These cells are going to be well suited towards phagocytosis and just general mayhem and destruction because they have all these vesicles filled with something. In the case of neutrophil, it's going to be acids, Basophils are going to have lots of histamines and things, so they have some kind of compound that they utilize to carry out their job. Monocytes and lymphocytes function a little bit differently, and we'll talk about those. Um, so the most common granulocyte is the neutrophil. It's the most common white blood cell, period. But 50 to 70% of all white blood cells actually are neutrophils. And these are kind of your traditional magic school bus white blood cell. They are the bacteria slayers. They phagocytize bacteria, which means they engulf bacteria. They wrap themselves around and endocytose or phagocytize the bacteria. And then once that bacteria is encapsulated within the cell, vesicles filled with acid merge with that vesicle containing the bacteria and release their contents and then digest and just degrade that bacteria. They can also do something called a respiratory burst, which is they release basically acid into the area where the bacteria are, so they can actually kill bacteria without phagocytizing them. And they even have, and this is just when it gets pretty metal, if you ask me, is they have spears. They have actual spears that they poke holes into the bacteria with, so that the bacteria die. So if you have bacteria, the neutrophils are going to handle that. Eosinophils are much rarer, 2 to 4%, unless you have some type of parasitic infection, in which case you'd have a whole bunch. My good friend, actually the best man at my wedding, he took his wife to it's somewhere out in the goodness, I don't know Polynesia, Indonesia. It's where they have the huts on the water. Is that Bora Bora? What is that? I don't know. Tahiti? I'm not sure. Anyway, he went there, and when he came back, his wife had both dengue fever and West Nile virus. And dengue fever is a parasitic worm, I believe. Anyway, and I think one of the tests that was probably pretty off was her eosinophil count, if it is indeed a parasitic worm. I think it is. One of you guys can correct me if I'm wrong. But anyway, so these address parasites. Basophils are very rare unless you're in the site of an inflammation response. So if you scrape yourself and it's bleeding, there's blood involved, because remember, red blood cells are in blood. They're not in your tissues. They're going to release histamine, which is going to bring in all sorts of inflammatory. It's going to mediate an inflammatory response, which brings in fluid and other things that will help help prevent infection and help address an infection that's already present. Agranulocytes are just lymphocytes and monocytes. Lymphocytes are actually very common. They're 25% of all white blood cells, but they're very rare in the blood, which is unusual for a white blood cell. And that's because lymphocytes actually live within your lymph system. Lymph is a really, I wish we could cover the lymphatic system, but basically your circulatory system is a closed loop, however it's leaky. So you have almost like a secondary circulatory system, but I hesitate to call it a circulatory system because it is not a circle. It's not a loop. Instead, it's a one-way system that brings fluid back from your tissues back to your heart. There's a little, I believe it's called your thoracic duct, where your lymph empties back into venous circulation right above your heart. Anyway, so it's constantly pulling fluid. If you've ever walked around Disneyland all day and your ankles are swollen up, your lymphatic system is working overtime trying to bring that fluid back. As it does that fluid, it moves through the lymph capillaries and the different collecting ducts, and it passes through these structures called lymph nodes. And these are nodes packed with lymphocytes. And there's different types of lymphocytes. There's memory T cells, there's naive T cells, there's cytotoxic T cells, there's B cells, there's natural killers. So there's all different types of lymphocytes, and they all do different things. But they're very rare in the blood. If you see them in the blood, there's an active situation going on, or different cells migrate from different areas. So immature T cells will migrate to the thymus to mature. So there are times you'll find them in the blood, but they're relatively rare. Lymphocytes are really the part of your adaptive immune system. You have kind of your innate immune system, which is your initial response to a pathogen. Neutrophils are going to play a huge role in that, various other types of cells. But then eventually, if that pathogen is not destroyed by those first-line defenses, lymphocytes get involved, and particularly in the form of producing antibodies. And antibodies circulate throughout your blood and destroy that pathogen. Then you have cytotoxic T cells, which destroy your own cells that are infected by the virus, and it's really complex, super interesting. Sorry, this quarter we didn't get to it. We're actually a couple chapters away, but so my apologies. But anyway, so that's lymphocytes, 25% of white blood cells, but rare in the blood. Then we have monocytes, about 3 to 8% of white blood cells. Monocytes themselves don't do a whole lot. Monocytes are basically inactive macrophages that are circulating in the blood, looking for a reason to leave circulation and engage in some type of warfare with bacteria. So basically they leave the blood and enter the tissue, areolar connective tissue in particular. You'll see a lot of macrophages. These are big phagocytic cells. That's why they're called macrophages, and they're highly phagocytic. If you ever popped a pimple, you've probably sent, millions of, hundreds of thousands of macrophages to their doom. They are fighting bacteria within your connective tissues, and your areolar connective tissue in particular, right under your epidermis. Anyway, and those are your white blood cells. Leukocyte disorder. Okay, so leukocyte disorders. So we have 2 listed here. Leukemias are just a category of cancers that cause a massive overproduction of white blood cells. Now, of course, this is very dangerous just due to it being a cancer, but also these white blood cells are not functional. So you have a bunch of non-functional white blood cells in your blood, which is where the name comes from, leukemia, which literally means white blood. So this is a family of cancers, and has some pretty unfortunate mortality statistics. I mean, there's different types, but generally speaking, that is one of many things that we talk about in this class that you do not want to get. Infectious mononucleosis is relatively benign. It's actually super interesting, though. It's caused by a virus called the Epstein-Barr virus, which is a virus in the herpes family. And what's interesting is that by the time we're 40, about 90% of us have it. If we were to look in our blood, we would see that there is this virus in our blood. So most of us at this point probably have this. But very few of us will ever show symptoms, and if we do, it would only be in our adolescence or young adulthood for whatever reason. I'm sure we know the reason. I don't know. I would really need to take a look at the literature to tell you that. But it gets its name due to the abnormal amount of monocytes that would be present in your blood. There's also an abnormal, kind of atypical type of lymphocyte that you would see circulating in your blood that you normally wouldn't see, and there's some abnormal antibodies that you might see. It's more or less, like any other virus with fever and aches and other symptoms. It can be very contagious, although it kind of has a mythology about it of being just incredibly contagious. It's actually less contagious than the common cold. It is spread through saliva, which is kind of why it's called the kissing disease, but really I think that has more to do with when mono symptoms typically present, which is in kind of young adulthood during that time, right? So the fact that it's spread through saliva in young adults kind of has that reputation, but lots of diseases are spread through saliva. It's just that this one happens to present itself in young adulthood. Anyway, so that's it's not listed here because it's a virus, because any virus would cause elevated leukocytes. It's a specific leukocyte disorder because of some of the things that are associated with it, like increased monocyte count, atypical lymphocytes, and atypical antibodies. So that brings us to platelets. Platelets aren't a cell, they're just a fragment of a very large cell called a megakaryocyte. Platelets are essential for the clotting process. They release thrombopoietin, which we'll talk about on the next slide. But basically, we have a hemocytoblast, which develops into a megakaryoblast, a promegakaryocyte, a megakaryocyte, and if you see these little extensions here, we have a special type of capillary in the red marrow called a sinusoid capillary, and it's very leaky, it's very porous. And these megakaryocytes kind of reach these arms into that sinusoid capillary, and these fragments break off. And as soon as that cell membrane fragment breaks off, it reassembles into a closed unit due to the nature of the phospholipids, which we discussed in the very first lecture last quarter. And they're these tiny little cell fragments. They're just floating around in our blood. So these come into play during hemostasis, which is the process that results in coagulation. So let's just run through these steps really quick. It's such a cool picture right here on the bottom left. So anyway, so step one, if in this case it's a capillary that is, or an arteriole, I'm not sure, that is torn. Okay, so step one is vasoconstriction. A vascular spasm occurs, the smooth muscle contracts, and that causes vasoconstriction. That reduces the blood flow through that area, which reduces the blood loss. Step two relies on the exposure of collagen. When a blood vessel tears, there's going to be collagen fiber present. Anytime there's an injury and tissue is torn, connective tissue has a ton of collagen in it. Blood vessels have collagen, so there's going to be exposed collagen fibers. Well, platelets just happen to stick really well to collagen fibers, so the platelets adhere to the collagen. Once that happens, the platelets release chemicals that make other platelets more adhesive. So those initial collagen fragments get covered up by platelets, the platelets bind to them, but then those platelets become sticky. So other platelets start to stick to them. So you start to get this coalescence of platelets in this tear. That leads to fibrin coming out of circulation. It's fibrinogen, it's dissolved in the blood, and then when it's exposed to these platelets, it comes out of blood, it comes out of solution and becomes a physical fiber called fibrin that forms a mesh. That mesh then starts to catch the red blood cells as they're leaking out of that tear. So the platelets, the fibrin, and the white blood cells form this little coagulation, this little scab, basically, this little clot that keeps the blood from leaking. And then over time, that's remodeled and repaired. But this initial coagulation is what stops the loss of blood, which is the priority. And that is it. That really brings us to really our last thing here, which we have a few slides for, that I want to kind of slow down and spend some time on, and that is transfusion reactions. So transfusion and blood replacement. So we're going to be talking about the ABO blood typing system, and very briefly the RH groups. And I really want you to understand what types of blood would react with one another, but more importantly, why. So let's get right into it. So blood typing is based on what type of antigens are present on a red blood cell. So your plasma membranes have highly specific antigens, so glycoproteins, on their external face. So any glycoprotein on the surface of a plasma membrane is an antigen. In red blood cells, we call these specific antigens agglutinogens. For my sake, because saying agglutinogens and agglutinins over and over is not only confusing for you, but for me as well. We'll just continue to refer to these as antigens. But just know if you see the word agglutinogen, that's referring to this specific type of antigen on red blood cells. And it's because these antigens are involved in agglutination reactions, which is when antibodies basically destroy red blood cells in the case of a mismatched blood transfusion. So that's why they're called agglutinogens, but we're just going to call them antigens for simplicity. There are a lot of groups of antigens, 30, but ABO and RH are the most important. If we match these, the likelihood of a transfusion reaction is basically 0 if the ABO and RH groups are matched. It's not basically 0, it's low. The chance of this happening is very low. Okay, so let's just talk about, do we even talk about RH? Do I have a slide? I do, okay. But let's talk about ABO blood typing. It's based on the presence or absence of two antigens, type A antigens and type B antigens. So really quickly, let's just run through it. If you have type A antigens on your red blood cells, your blood type is A. If you have type B antigens on your red blood cells, your blood type is B. If you have both, your blood type is AB, and if you have neither, your blood type is O. So you can kind of think of 0, because there are none. Neither A or B antigens. Okay? The type of antigens that are present on your plasma membrane will tell us what type of agglutinins are present. And agglutinins is a complicated way of saying antibodies that pertain to these antigens. So let me explain. If you have type A blood, you have anti-B antibodies. So if you got a red blood cell, if someone gave you type B blood with red blood cells, your antibodies would attack that blood, it would clot up, and it could cause you a lot of trouble. You don't want clotting occurring within your blood all over the place. So if you have type AB blood, you have anti-B antibodies. Probably write this down if you have paper handy. So if you have type A blood, you have A antigens and anti-B antibodies. If you have type B blood, you have B antigens and anti-A antibodies. If you have A and B antigens, which antibodies do you think you have? Well, think about it. You can't have anti-A antibodies if you have type AB blood, because your antibodies would attack your own cells. Because if you have AB blood, you have A antigens. So it wouldn't make sense for you to have anti-A antibodies. But it doesn't make sense for you to have anti-B antibodies either, because you also have B antigens. So if you have type AB blood, you have both antigens and you have neither antibody. So you have no type A, I'm sorry. You have no anti-A antibodies and you have no anti-B antibodies. So just remember, no antibodies with AB. Type O blood has no antigens, and therefore it has both antibodies. It has anti-A antibodies and anti-B antibodies. Okay, I'm going to skip down several slides here, and then I'll come back. This is so crucial, this particular, I don't know why I can't click it. Here we go, let's highlight it. This particular right here will be necessary for you to memorize. Very rarely do I say you have to memorize things verbatim, but the recipient's antibodies, so put antibodies there. Agglutinins is just a more specific word, but it's a type of antibody. Recipient's antibodies attack the donor's antigens. So write antibodies, and then over here write antigens. The recipient's antibodies attack the donor's antigens. That is totally necessary for you to understand transfusion reactions. But before we get there, I'm going to go back to where we were. Okay. Okay, so it's all here. Type O blood, it's already written down. I just told you to write it down, it's already written down, I'm sorry. Type O blood contains anti-A and anti-B antibodies. Type A blood contains anti-B antibodies and vice versa. Type AB blood contains neither. All right, then we have RH blood groups. So there's 52 named antigens, RH antigens. Each of these is called an RH factor. Only 3 of them are common. If you have any of them, you're considered to, your blood type is going to have a positive after it. It's RH positive. If you have none, it's RH negative. So you could say O negative, O positive. So we'll explain how that might come into play with transfusion. Actually, no, let's just keep it simple. So we're just going to talk about ABO transfusion reactions, but understand the same principle applies to Rh blood typing as well, where the recipient's antibodies attack the donor's antigens. I just want to get to putting that concept into practice right away. So here we go. This is a great image that illustrates the antigens and antibodies present in each blood type, in the ABO blood types. So let's say we give type A blood to someone with type B blood. So the person with B blood has surface antigen B, okay, and it has anti-A antibodies. So if you give A to B, what happens? The recipient's antibodies, right here, anti-A, would attack the donor's antigen. This drawing is getting silly. Forget the drawing for a second. If I give A to B, the anti-A antibodies in the type B blood will bind to the surface antigens on the type AB blood and cause agglutination, which this image is kind of demonstrating here. It's clumping of cells and then lysing of cells. This is not good. You do not want this to occur in your blood. So if I gave type A to B, anti-A antibodies would attack the surface antigens on the donor's blood. Let's switch it up. If I gave B to A, anti-B antibodies in the recipient would attack the donor's B antigens. The recipient antibodies attack the donor's antigens. Now, you might be thinking, why does he keep mentioning that? Well, because technically, again, let's say I give type AB blood to, I keep saying type AB. Let's say I give type A blood to someone with type B blood. Remember, the recipient's antibodies attack the donor's antigens. Well, so what do these antibodies do? So that's the thing. They still bind to the surface antigens and cause agglutination and lysing. The thing is, you're giving a relatively small amount of antibodies, and when you're transfusing blood into somebody, you're giving a relatively small amount of antibodies. By volume, you're giving quite a bit of red blood cells. So the agglutination of these red blood cells is bad. This relatively minor amount of agglutination caused by the donor's antibodies reacting with the recipient's antigens is relatively minor. Now, in this case, it doesn't matter because either way, if you give A to B or B to A, you're in trouble. You're losing your job and you're probably getting sued, or your hospital is, or whatever. So that's not that important, but it does come into play when we talk about AB. So let's say I give type AB blood to type B blood. Okay, is that cool? Remember, the recipient's antibodies attack the donor's antigens in a dangerous transfusion reaction. So in this case, the anti-antibodies would bind to the surface A antigen on type AB blood, so the recipient's antibodies would attack the donor's antigens and you'd be in trouble if I gave AB blood to B. But get this, if I gave B blood to AB, so I'm just switching, instead of AB being the donor, now AB is the recipient. That's okay. And you might say, well, how can that be? The anti-A antibodies are going to react with the surface antigen. But remember, in a transfusion reaction, it's the recipient's antibodies that react with the donor's antigens. Well, guess what? In this case, there is no antibodies. So type AB blood does not have anti-A or anti-B antibodies. That makes it the universal recipient. You could give type A blood to type AB, type B blood to type AB, type O blood. It doesn't matter, because remember, in a dangerous anytime there's going to be a transfusion reaction, it's always going to be the recipient's antibodies reacting with the donor's antigens. There are none. So type AB blood is the universal recipient. So that's why I want to illustrate, because again, if you give either one of these blood types, so type AB, there will be a minor amount of agglutination because there are anti-B antibodies in the donor's blood. There are anti-A antibodies in the donor blood. There's just there's not that many of them in that volume of blood to cause a lot of damage. But if we reverse this situation and give AB blood to B, every single one of these red blood cells that I'm giving this donor is going to agglutinate, because collectively in this recipient's body, there's enough antibodies to agglutinate every single donated red blood cell. The opposite wouldn't be true if I gave B to AB. AB is the universal recipient because there are no antibodies. There are no anti-A or anti-B antibodies. Okay, and that just leaves us with type O. So let's say I give type A blood to type O. What's going to happen? Well, the recipient's anti-A antibodies will react with the A antigens. Let's say I give type B blood to type O. What's going to happen? Are you keeping your job? No, you're losing your job because the anti-B antibodies react with the surface B antigens on the donor's blood. Let's say I give type AB blood. This is all bad. We have the anti-A antigens reacting. We have the anti-B. I said antigens. We have the anti-A antibodies reacting with the anti-B antibodies reacting with the surface antigens. So if you're type O blood, you can only receive type O blood. But guess what? You're a hot commodity for the Red Cross because type O blood has no antigens. And remember, in a transfusion reaction, it's the recipient's antibodies reacting with the donor's antigens. Well, guess what? The donor doesn't have any antigens. So type O blood is the universal donor. You can give type O blood to any one of these. There will be a minor amount of agglutination due to the antibodies present, but there won't be a highly dangerous transfusion reaction because there are no antigens. And remember, again, it's the recipient's antibodies that react with the donor's antigens in a transfusion reaction. Okay? Hopefully you're not super confused. If you can't get it conceptually, this might be one of those times where you just memorize. AB is the universal recipient, so that's easy. O is the universal donor. You can't give A to B, you can't give B to A, right? So from there it's pretty easy. You can probably get most of the questions, if not all of them. But it is always nice to understand it conceptually. So I hope I got you there. Okay, so let's move on. So transfusion reactions occur when mismatched blood is transfused. We just went through that. It's the recipient's antibodies that attack the donor's antigens. O is the universal donor. AB is the universal recipient. And that, my friends, is the end of Chapter 17, Blood, the River of Life. Thanks for listening. Talk to you soon.